I am honored to pass the stage to Dr. José Padron, full professor at the University of Laguna, Tenerife. He founded and now leads the research group Biolab at the University Institute Bioorganica Antonio Gonzalez. His research focuses on anti-cancer drugs, with particular interest in hard-to-treat cancers. His research involves the early pharmacological profiling of small molecules with anti-proliferative activity. At Biolab, he tries to shed light on the mode of action by studying the effects of the products at a molecular level. His ultimate goal is to identify the biological targets at which the active compound is directed.
Thank you very much for your kind introduction and your invitation to present the results we have obtained with this new module of CXA, the cell death assay. As an introduction, I will like to show you that we are a research group located in the Canary Islands at the University of La Laguna. Our main research is related to the pharmacological profiling of small molecules, in particular for the treatment of cancer, but we have expanded our capacities also to molecular modeling, to synthesis, or even to study the natural products coming from our endemic species. Our main goal is to get molecules that are able to be delivered to patients for the treatment. NanoLife CXA has been, for us, revolution in terms of how we can see the results, and with the help of my talented and enthusiastic students, we have been able to produce some results that we will show you in the next slides. Here I would like to present some compounds that certainly I will not show the structure because of confidentiality agreements with the producers, but we can see that in the treatment of this non-small cell lung cancer cell line, the compounds are able to induce necrosis. So the system is telling us that cells are dying from the very, very beginning. We can see on the right side of the screen the graphs that shows how fast the percentage of those cells increases in all the treatments. These very nice blue colors are transformed into data that we can use for our presentations or for our publications, and it's telling us about how the compounds are interacting in time. Certainly there is no manner to see apoptosis coming here because we have record this experiment every 3 minutes, and the system is just showing direct necrosis from the beginning. We can see the percentage of cell death, but we can also give the data in terms of percentage of necrotic cells from the total of the population. This result, which is really, really interesting, it makes it even much more impressive when we go to the next slide and we see what happens with a similar non-small cell lung cancer cell line, the A549, when we treat them with the compounds. As you can see in the video, the astonishing effect that we are observing in the previous one is hardly happening here. We see that cells are almost inert to the treatment. How can we distinguish this? Well, we see here on the graph at the right side of the screen that the percentage of those cells is really low. And in fact, one of the compounds is not even able to produce any cell death at all. Another very important feature that we would like to show here in terms of this screening in this particular setting is that we are able to understand what happened to healthy cells. So are they stressed? Are they alive? Are they in good shape? And this is what the machine gives us as the health cell index. So if we can see here, we observe that cells that are not dying are perfectly healthy. So in fact, this is the first time that we are able to distinguish subsets of population within a single cell line. This is something that could not be observed before in regular flow cytometry experiments, for instance. So we know exactly that our compound is killing some cells, but those that are not being killed are not being disturbed by the compound, which is a very, very interesting point to start for research. In the next sample, I'll show you two compounds that's supposed to interact with the mitochondria. So those mitochondriotropic compounds are able to induce cell death in these HeLa cell lines that we use as model. As you can observe from the images, we start to see that the cells become less healthy, they start to bubble, and then at the end, they die. More importantly, how can we translate this into usable data? We have this graph. So we observe on time how cell death is occurring. So we know how much percentage of the population is dying as the treatment is going on. In fact, very interestingly, we can have a cell death rate. This is something that is impossible to get from conventional experiments. So for the first time, we are able to include the variable time in our experiments, which is going, and I anticipate, is going to produce a lot of new information. It's going to change the way we are looking to experiments. Certainly, with this new module, the cell death assay, we are gaining a lot of information that are going to help us to distinguish particularly the mode of action of all these new compounds, and we want to expand also to other standard anti-cancer drugs to revisit the known mechanisms of actions reported in the literature. Very interesting in this slide that we have the same compounds and the same cell lines is that we can get an outcome of the experiments in different kind of plots that are interesting to see at different levels. So for instance, here on the right side, we see that we can plot the percentage of living apoptotic and necrotic cells all together. And in this particular experiment, we have a clear view that compound X and compound Y have a different behavior among time. Those compounds have the same potency in all the experiments we have tried before. Those compounds behave completely similar in all the experiments we have run before. But this is the experiment where we see there is a difference between the two of them. So one compound is acting faster than the other. What is the reason for that? Well, that's the good question that this new system is going to start to make us. So we will have to revisit all our experiments and perhaps include the variable time in this new way of looking at the cell death or cell intervention by the treatment of drugs. And this is all for now. I would like to thank all my students, in particular Adrián Puerta and Adai González, who helped me in the acquisition of all this data, and certainly NanoLife for helping us and letting us try this module, which certainly is going to be our top priority software in the lab. Thank you very much.